So hello everyone to this lecture on workflows on GDPR compliant platforms. VRE and EBRAINS are the two platforms that we are going to talk about today. And what is this goal or one of the main goals of these workflows? And just to give you an impression and repeat a few things that you probably already have seen in other talks. So the goal here is basically to construct brain network models from MRI data. And we have different types of input data for these pipelines. T1 weighted data, which gives a nice structural contrast. Diffusion weighted MRI, which is used to estimate the connection strengths and time delays of interaction between brain areas. And then we have fMRI and fMRI networks, which indicate about functional interaction between brain areas. And again, basically the brain is parcellated into a set of regions, and then these fiber tracts are reconstructed. And from these we have then matrices like the weight matrix that indicates the coupling strengths between different areas, and the tract length matrix. And these are the input for a brain model and for simulating the brain. And simulating the brain is also one of these workflows that we will look at. And just to quickly make this distinction. So we have on the one hand the structural connectivity, which is the input for the brain model. So these are these fiber bundles. But then we are looking at the dynamics that emerge from this structural skeleton across which the neural population models interact. And this is then other types of connectivity that emerge from this. For example, functional connectivity, which is the network interaction, the correlation between brain areas over longer times, and then also dynamic functional connectivity, which is the interaction over shorter times, and also effective connectivity, which is basically the causal or the directional effect as we can study it with brain models. And the output of such neuroimaging pipelines that we use to construct brain network models are then functional connectomes, for example, structural connectomes, projection matrices that allow us to project the activity that is simulated on cortical surface into the sensor space, specifically EEG or MEG sensor space. Then we have region-wise fMRI data, which we can also compare them with the empirical fMRI data. We also have surface triangulations for creating volume models that describe the conduction of electromagnetic fields through these different tissue types in order to project the neural activity into the sensor space in order to compute such projection matrices, for example. And we have brain maps, which are atlases or parcellations of the brain into a set of different regions, which we try to do in a way such that it is comparable across different persons. So if we are now in this temporal lobe in one person and it is a certain function, then we would also like to identify the same functional region in other people. And this is an important precondition for constructing brain models. And what are typical steps in such pipelines? So often the imaging data contains artifacts, distortions that need to be corrected. So computer vision algorithms and image processing algorithms that basically detect and correct these artifacts, and also surface reconstruction, basically in order to have 3D models of brain structures. Cross-Modal Registration is basically the registering, the alignment of different MRI imaging modalities or also other imaging modalities to bring them into the same space, to have the same parcellation, the same atlas, and to have different types of data, like different fMRI contrasts, for example, comparable. And segmentation parcellation is the process in computer vision and image processing, basically where different structures in an image or a volume are segmented, are isolated and delineated. Warping and alignment is also standard image processing algorithms where different structures that are depicted in images are brought into alignment in order to have them comparable. And signal aggregation is also a typical step where the signal is made simpler, so to say, in order to in order to better analyze it, basically. And this is just one exemplary sequence, so we don't need to go through all these steps. The message here is that basically these pipelines consist of many steps, often also similar steps that are repeated at different positions, and that basically need to be coordinated in the form of a workflow that ensures that from the input data over the different processing stages to the output data, no errors or implausible processing steps occur. And this is basically not so easy from different perspectives. And we will look at these perspectives in the following. And here's just a quick example that you get a visual image what a typical operation is. So here we load a T1 image where we can nicely see the structure, and here we extract the brain. So this is already quite a problematic operation to just segment or delineate the brain. Because as you can see here, the brain looks only marginally different than the surrounding structures. And here we can see that the skull strapping actually didn't work so nicely. So we have to play around with parameters. So these workflows are very complicated. This is the message basically. And it's a big task basically to have these workflows reproducible and also to use them for different data sets. And to solve this problem, basically, there are now great efforts that bring researchers together in cloud infrastructures where they have high performance computers and storage and also shared spaces where they can interact and perform processing operations together. And EBRAINS is one of these platforms that offers different kinds of services like data and knowledge, brain atlases, brain-inspired technologies like neurorobotics, medical data analytics, and also simulation services, which is basically what we are doing, the virtual brain and the virtual brain pipelines. And what are these services, these virtual brain services at EBRAINS? So on the one hand, we have the core brain simulation functionality of the virtual brain. Then we have the multi-scale co-simulation that you also saw in other lectures. Then we have specialized algorithms for high-performance computers that make use of the parallelization, the heavy parallelization, for example, of high-performance computers and also of their specific hardware. There are also certain parameter fetching models implemented that can be used to reverse engineer parameters, for example, of epileptic patients and their epileptic target and propagation zones. Then we have the image processing pipeline, which we will talk a lot about, which is basically a prime example basically for these kind of workflows, because this image processing pipeline integrates a lot of different aspects and a lot of different problems come together there and are then addressed there. And then we have TVB ready data, which is data that is ready to be simulated. So let's have an overview over the TVB on EBRAINS cloud infrastructure. So we will go to some of these aspects in more detail, but let's just give a brief high-level overview. So these cloud platforms and this kind of research, brain research, makes use of personal health data. So we always have data from human subjects, and this comes with certain privacy concerns. Because we will see later what is the reason, basically, because this data is very rich and can be used for wrong purposes, so to say. And therefore, a lot of efforts have basically to be done in order to protect the privacy of this data, like end-to-end encryption, like sandboxing, we'll talk about this later, and also identity and access management. making sure that only the right persons can, or the persons that have a lawful basis can access the data. So there is a certain security gap basically that aims to isolate this sensitive data from exposure to any number of people. So this data needs to be protected. And then we have different core services in eBrains that are heavily used and reused by different cloud services of this platform. We will go through each of them individually, and then these are all connected via RESTful API and Unicore, which we'll also talk a bit more about. And these basically integrate now all these different deployments and variants of these 6 workflow services, TVB services, that I mentioned earlier, and which we'll look at in more detail. This is also, all these services are also connected with each other and with other higher level services of the eBrains platform, like for example, the neurorobotics platform, which simulates and controls virtual robots. And also Zebra, which is an interface for the multi-level human brain atlas, which resolves cortical structures with unprecedented resolution. So one major component of eBrains is the wiki or the Colab space. It is a space basically where teams can work together, where they can create documentation, where they can exchange and basically organize the contents of research. Likewise, Drive is also integrated with the Colab, or forms one part of the Colab. And Drive, as the name indicates, is basically a online file system, very similar to a local file system where you can just organize, store, read and write files. You can do the same here in these Colabs, with the difference that you can do it collaboratively with your team, basically. And you can basically add people to these teams. You can make them public or private as needed. Then the eBrains Lab is a space where Jupyter Lab instance is hosted. So every eBrains user has their own container, basically, where they can run their own Jupyter notebooks, Python notebooks, or R notebooks, and so on. Can run these notebooks and can also share them the notebooks or the data or the outputs with others via this shared file system. Also Office applications. So if the research is done, you can also collaboratively write up the paper. And then OpenShift is a container orchestration platform, and has all kinds of services for deploying containerized services in a cloud infrastructure, basically. So all of these services are in a container orchestrated by Kubernetes or OpenShift, which is a platform basically that makes sure that these containers are executed when free resources are available, that moves these containers to the free resources. So this is a load balancing, basically makes sure that always the consumable resources are used for the jobs that are there, and also logging and all kinds of other cloud services are provided there. Then eBrains is connected with a network of European supercomputers, the Phoenix supercomputing network. Hello. Can you mute yourself, please? Thanks. So we have these European supercomputers like Jülich, CSCS, CEA, Barcelona, and these provide high performance compute powers to the platform, basically, and make the goal is to have a seamless experience where you as a eBrains user have a joint platform that basically works with these federated infrastructure services. So we have all these supercomputers at your back end, and they are federated via a common layer, Phoenix, to make the experience easier. So we have different interfaces to connect these different services. The RESTful API principle is used throughout the infrastructure. So basically, one sends short code snippets, so to say, between the local computer and the cloud. And this can be done to exchange structured data and metadata and data about data. And all these workflows can also be automatized. So there is a exchange of secrets and identifier tokens that basically allow to communicate machines with each other in a secure manner, such that the data is still protected and only authorized clients or users can access the data. And basically on top of this sits then Unicore, which is also an API basically, and this time for the supercomputer. So Unicore basically exposes an API to the user that makes it easy to operate a supercomputer from the Jupyter lab. So if you would directly connect to a supercomputer with a Linux shell session, for example, you can here have similar commands and basically operate the supercomputer or automatize the operation of the supercomputer from a Jupyter Notebook. So let's go through some aspects of these different workflows. So the first one is the virtual brain, the brain simulator. Now there are many different ways how you can use the virtual brain, either locally or in the eBrains cloud infrastructure. So locally, you can just download a compiled version and just use it locally on your computer. Similar also with the Docker image. So you can pull this Docker image onto your computer. Yeah, it's platform independent, so it really just needs the Docker runtime engine or Singularity and can be used there. Then we have the web GUI, which is then really a true cloud interface. So you can reach this via this URL, and then basically don't need to install TVB at all in your computer. You have it in the cloud, and you can run it in the cloud. And if there are resource-intensive jobs, they are even executed then on a supercomputer without having the user to perform all these steps manually, basically, and to install TVB at a supercomputer, upload the data in a secure way to the supercomputer, perform the processing, and so on. So this is all automatized there. We also have Python notebooks, which you can then directly run in the eBrains lab, in the Jupyter lab, or you just install the TVB Python library and can then use it from every Python script. And one aspect of this is also that you can get TVB-ready data from Knowledge Graph. So Knowledge Graph is also another important component of the eBrains infrastructure. It allows you to basically search through all the metadata that characterizes the data sets that exist in eBrains, and that you find them, that you request access to them, and use them basically in your own research. And then also, of course, output your own research outputs and integrate them into the knowledge graph. The knowledge graph basically shows you for each data set, such a data set card where you get all the metadata information, all the data about this data set that you need in order to understand and use the data set. For example, a verbally written data descriptor for humans to read, publications, but also there is machine readable metadata basically that sits behind what we can see here and characterizes exhaustively such a data set. So just to show you that it's really very easy, if you want to try it out yourself and want to simulate a brain there, you can just open this URL and go to the TVB web interface, select the deployment site, so in Switzerland, CSCS, for example, a supercomputer, or Jülich, and then directly head over to the simulator. You can just use the default connect home that is there in a test project. You would just use the default parameters that are there and can directly launch a simulation, wait until it is computed, then you will see the result on the right. A time series was produced, and then you can visualize these with a number of different visualizers, for example, the 3D brain activity visualizer. As mentioned, alternatively, you can run TBB locally with a container image, or install the TBB Python package with these simple commands and directly start simulating. So these few commands basically already set up a full simulation, yeah, which you can use to simulate brain activity. Okay, now let's move over to the TBB image processing pipeline. So we have, yeah, here basically we would start with a few words on data privacy. So this is a big aspect of these pipelines, as mentioned, is privacy. We have, so scientists all over the world, researchers, they create huge amounts of valuable health data from healthy subjects, also from patients. There are many people who donate their data to science, basically, and make it available such that it can be used for research. But the problem is that these data are so rich that openly sharing and processing this data on shared infrastructures can severely compromise the privacy of research participants. And the reason is that these data sets are really rich. So we have basically not only fingerprints at our fingertips, but basically all over our bodies. So every small millimeter voxel of such an MRI data set basically contains such a wealth of information that they cannot be easily pseudonymized, which was one approach. So the idea was to just remove all directly identifying information like names, addresses, birth dates, and so on. But this is not enough because, or faces, as we can see here, a face is removed from the MRI image. But this is really not enough because these data contain so many information about a human, which is obviously also the goal, because this information shall be used for science, right? So that it can be also misused. And as you can see here, for example, in a fMRI data set of a human, we can read really a lot of highly private information, I would say, like with a large correlation, we can already infer their fluid intelligence and other, I would call them private information, right? So this can be inferred from human subjects, and this is not only theoretical, but this really happened. So, for example, there was the Human Genome Project some time ago, where the full genome of several individuals were sequenced. And then a few years later, there came these genealogy websites where people posted snippets of their own genomes and also their surnames, for example, other personal data openly on these websites. And then these researchers basically just combined these two different data sets, and this allowed to reverse engineer the surnames, the identities of the Human Genome Project subjects and also of their relatives, basically. And this basically gives very detailed information about the genomic setup of a large fraction of the population. It has already happened, right? With very simple, I would say, data at this time. Now it's much more complex and much richer. So it's very easy, basically, to re-identify humans based on very little information and to reverse engineer highly private information. So this, obviously, this comes with technological change, with the internet, with our new imaging modalities, with computing infrastructure, and so on. So the legislature, the law, was not really fit for this kind of world, basically. So in the European Union, we now have the General Data Protection Regulation, which addresses this problem, basically, that we now have a wealth of information that can be very easily shared and misused. And these set of regulations basically protect citizens from having their data misused. And there are different entities or persons or groups of persons under GDPR. So we have on the one hand the data subject. This is the person who donates the data. Then we have a controller, and this is really someone who controls the data and therefore has the responsibility. And to become a controller, you don't have to do anything, right? So if you snap a photo with your phone, then you are and there's a person on it, then you are a controller and you control personal data in this moment. Or if suddenly a piece of paper with health information of a person appears on your desk, then you're also a controller. Or if someone sends you an email with health data, then you're also a controller. And with this comes a lot of responsibility. A little bit less responsibility has a processor who only acts on behalf of the controller and who have a formalized relationship. But the controller really has the responsibility to implement appropriate technical and organizational measures to ensure and demonstrate that processing is performed in accordance with GDPR. And how can the controller do this? This is, for example, clarified in Article 25 that by default, personal data are not made accessible without the individual's intervention to an indefinite number of natural persons, and that controllers and processors shall implement technical and organizational measures to mitigate risks like pseudonymization, encryption, resilience of systems, availability restoring, testing, adherence to code of conduct, certification, and so on. So basically, the controller has to do everything in their power to protect the data and to demonstrate this also. Otherwise, data subjects can basically get the right in a European court and can basically veto their processing of their data. So there are different supervisory authorities in the EU, but also on a national level, data protection officers or federal data protection entities that basically ensure that the processing of data in the jurisdiction follows these processes basically. And all of these different entities need basically to be aligned and need to be integrated, need to communicate together to really ensure the compliance with GDPR. And the technical infrastructure for this is not really existing, but we are working on it, or it exists partially, but there's a lot to do basically to really comply with GDPR. The thing is that health data, which is our primarily of primary interest for us, is generally prohibited. So the processing of health data is generally prohibited. So there is no such thing also as pseudonymity. You cannot say, my health data, I have removed all the names and so on. So I can now just put it on the internet, make it available for download and stuff like that. This is not legal. There is no pseudonymity basically, because if you can basically reverse engineer or re-identify these persons by bringing it together with other data sets, then there is no pseudonymity. And this is with health data basically always given. And we cannot anticipate in which way, for example, artificial intelligence algorithms will be able to map different data sets together and reverse engineer identities. And therefore, health data is generally prohibited and may only be processed if the data subject has given explicit consent to one or more specified purposes. So the data subject must really be in the know what happens with the data and who processes the data. Another suitable lawful purpose is that the processing is necessary for scientific purposes in accordance with Article 89. And there we again end up with the technical and organizational measures that ensure the respect for the principle of data minimization. So we always come to the point that basically the controller has to ensure that the data is secure. And if this is the case, then if they can demonstrate that, then they must not fear prosecution so much. So let's have a closer look at the pipeline. So, or at the general ideas behind these pipelines. So the TBB image processing pipeline is a pipeline of containers. So the idea is that you have containers for different tasks. Like one task is, for example, take an MRI data set and compute a structural connectivity or perform the tractography that is used to infer the structural connectome. Another concern may be only the fMRI processing. So basically, ideas here to modularize the processing into a bit more digestible units. Of course, these different steps can be made finer or more detailed and containerized in a basically an almost arbitrary level. So you can also make 2 containers or 100 containers out of this basically. But this seems like a plausible separation. So we have one container that basically deals with the structural connectivity and the tractography. Then we have one container that deals with the fMRI processing, and then one container that brings the outputs from these 2 together and transforms them into a TBB usable format. And the pipeline has 3 main interfaces. So on the one hand, there is a shell script basically that you can directly run on the supercomputer that acts like a daemon or a client on the supercomputer that allows you to basically perform these pipelines on the supercomputer without needing to directly interact with the supercomputer. You can basically perform these steps with 4 or 5 commands basically. And this integrates not only then the necessary steps for data privacy, but also for data reproducibility, which we will come to later. The Jupyter Notebook is also a way to operate the pipelines. So here we use the supercomputer from a browser, from the Jupyter Lab in a browser, where we can have Python code, R code, and other code, Julia, for example, and automatize our processing steps or integrate them also with simulation steps, for example. And we also have a GUI, which is also integrated with the TBB Web GUI. It's on the connectivity tab. You have to select the third button, which I shown earlier from the TBB Web GUI, and there you can also operate the pipeline and specify these parameters basically from a GUI. So this is how it looks like. So here's this connectivity tab, image processing pipeline, and then here we can then select different parameters and just launch the workflow from the GUI. So what happens then? You configure the processing with the GUI, and then on the supercomputer, an orchestrator or daemon or client runs basically these containers, or first of all sets up the workflow and then runs the containers and stores all the provenance information with data-led. So the provenance information is basically information about the different processing steps on a very detailed level that allow to repeat the processing. So this is really a big problem basically for not only neuroscience but science in general. We'll come to that later, that many results are not reproducible, that even when the paper is, for example, very detailed, or even when the code is shared and also the data, then even then it doesn't work often, because there are tiny steps that are often just not mentioned. We'll come to that later. So here, all of these problems are addressed basically, and make sure that all processing steps are really captured. And then we can, after the processing happened, the GUI will notify and the results can be downloaded. Similar, we can do this with the Jupyter Notebook. So here we would interact with the eBrains drive. We can, for example, upload our data into a private repository. So eBrains currently is working on the sensitive on services for sensitive data. So this is the health data cloud. I will also briefly mention this later. So at the moment, basically, when you process sensitive data, you have to do this privately or with the lawful other data controllers that you have identified. And if you do this, you then upload the data using the drive and can then from the Jupyter Notebook forward them to the HPC, and then also from the Jupyter Notebook basically create these job descriptions for the supercomputer that allow to run these commands, and also then retrieve the results and download them first to EBRAINS, where you can process them either in Jupyter Notebooks or also then to your local file system to further process or share them. Now there are certain risks associated with this workflow with shared infrastructures basically, and with research that basically happens over open channels like the internet, for example, or channels that can be intercepted. So basically the internet and also these platforms like EBRAINS and so on, they all are based on logical separation. So the wires all transmit the data from everyone, right? But it's separated on a logical level, on a data or information content level, basically. And this, opens up the possibility for backdoors, for example, for hacks and for code that is not trustworthy, basically, that makes it possible that information is intercepted, that it leaks out, for example. So if there's an administrator in a cloud workflow or in a cloud platform, then administrators often have a lot of power, right? They can look into personal file systems often and so on. So all these things must be ensured that they don't happen, basically. And this is these are things that were not really considered when all these infrastructures were built. Like the internet wasn't built for this kind of private information. This is the inventor of the internet, Tim Berners-Lee, basically, he now dedicates his whole time on rewriting the protocols, like the IP protocols of the internet, in order to make them more secure and to make them fit for a world where private data is heavily sent around all the time. So these things can always happen. Also a break on device, so not only the physical device, like someone goes into a data center, which is obviously protected, but there are always people there, and there can always happen things. And also on a logical level, there are always backdoors discovered and always security risks discovered. Basically every week or every day there come updates for one of the hundreds of components that are used to build such platforms. So these platforms consist of hundreds of thousands of programs, basically, that are all orchestrated and work together. And there can be just one program that leaks data to the outside, which would then corrupt the whole workflow. Likewise for user impersonation. So if a secret that identifies a user is leaked, and this can also be used to corrupt the privacy of the data. And therefore, extensive measures need to be undertaken in order to protect the privacy of the data. For example, encryption, access control, sandboxing, and automation. So we'll go through these step by step. Encryption obviously is we make the data unreadable, so no human can understand this data anymore, also no machine. To crack this code, you would need extensive computing power. It's not easy. You cannot simply reverse this process. So this is a very important technology, basically, for ensuring the privacy of the data, and therefore requirement is that sensitive data is always encrypted by default and only decrypted during the actual processing. So only when the data is really needed, it may be decrypted, and then ideally also in an automated process that basically doesn't allow any leakage of data. For example, using sandboxing. So sandboxes, I would then encapsulate or containerize the sensitive data such that no viruses, for example, can access them. So when we can now combine encryption with automation, we can get even more secure. So we can or have set up a system basically that ensures the personal data is encrypted before it leaves the computer of the data controller, and also that the results are encrypted on the source system before they are being returned to the data controller. And these secrets or passwords or keys for decryption are always created only shortly before the processing and always stay basically on the target location. So if the target is the supercomputer, then the supercomputer produces a public and a private key, and these two keys work together. So the public key can be safely shared because it can only be used to encrypt the data, while the secret that is needed to decrypt the data will never basically leave this process and will always be kept safe there. And likewise for the other direction, we can reverse this process and send the data back to the user. And basically, then all processing results may only be written inside such sandbox file systems and securely deleted before termination. And by combining then encryption with automation and sandboxing, we get quite a secure system. So sandboxing, you know this from your phones. They always ask whether they have apps. Always ask whether they have the permission to, for example, access your photos or contacts and so on. So this is sandboxing basically. So there's only a controlled set of resources provided to a process, and ideally, or yeah, it is mandatory that this available resources are so small that the minimal small set of resources needed to perform the processing goal, and at the same time that it ensures that these resources cannot be used to harm the system or the data or leak the data. So these unencrypted personal data and secrets may only be written into a temporary file system namespace that is invisible from the host and fully and securely cleaned up when the last process exits. So Linux, for example, has extensive mechanisms to isolate namespaces, computing processes from one another. And the workflow looks then like this, basically. We have the data controller with the local data that they want to process on a supercomputer, and in between there's EBRAINS, which serves as a layer to manage and to secure this whole process. So we just log in at EBRAINS and just start the workflow, and then the workflow instantly creates a sandbox and pulls the code that is used for processing into the sandbox. So from now on, we only live in the sandbox, which is not written on a disk, which is not accessible by the host system, which is just isolated from the rest of the system. And now on both systems, on the data controller's computer and also on the supercomputer, we create these public-private key pairs. And then we exchange the public keys. So the private key always stays either with the data controller or with the HPC process that uses it, and only the public keys are exchanged. And with this public key from the supercomputer, the data controller can then encrypt the data, and this happens then also automatically, and uploads it via EBRAINS onto the supercomputer. And then only inside this sandbox are the data decrypted, the data processed, and also the results again encrypted to make sure that never any unencrypted data leaves the sandbox. And only after it was encrypted, this time with the public key that was obtained from the data controller, it can then be safely sent over open channels, and finally then only on the data controller's computer be decrypted. Okay, so we have, I think, like something like 5 to 8 minutes or so, and then it's time for a break, and we can also ask questions, of course, if you like. So I would, just to wrap this up, go over this workflow again from a different perspective. So we have just seen this on a on a technical level, now on a more intuitive level. So we configure this workflow with the GUI again. So here we can set the parameters of this workflow. So for example, for the first container, for the second container, for the third container, specify the input data and so on. And then basically the pipeline takes over and does the rest automatically. So when the user hit the launch button, then on the HPC, a public-private key pair for encryption of input data is created, sent to the local computer, and there the input data encrypted. Then on the supercomputer, datalet is installed. We will come to datalet after the break. And then also, so this is the solution for reproducibility. And also the actual workflow containers are pulled. The input data is uploaded, encrypted, of course, and then decrypted into the sandbox. And then basically Git Annex repositories are created to reproducibly store input, workflow, and output. So what this means, we'll come to after the break. It basically is used to store the whole processing in a reproducible way, storing every small detail basically to recreate this workflow, and also to make sure that data cannot be easily overwritten, for example. And also it enables version control, multiple copies, synchronization between repositories, content verification, and so on. So what this all means, we will come to after the break. And then the pipeline basically creates a configuration for the supercomputer. So the supercomputer also needs a specific data format to know how many CPUs are needed, for example, how much memory, for example, the input and the output location, and so on, the time that the job shall run, and so on. This is then all done automatically, and the workflow executed, the results encrypted, the status in the GUI updated, and the button for download activated. And okay, to mention this as a last point here. So basically what we are now trying to do is to package this in a more user-friendly way, such that users can, with a very relatively simple language, set together or combine their workflows, create their workflows, and then all these necessary steps for data protection and data reproducibility basically happen automatically. And this, the goal here is to automatize this process. Okay, so I think now is a good time for a break. So if you have any questions, please ask them now. So it seems there are no questions. So in this case, I would suggest, we meet back in at 5:25 in about 4 minutes. Okay, hello and welcome back, everyone. Welcome to the second part of this talk. Let me just set a timer for the time. Okay. So another aspect of TVB on eBrains that I want to briefly mention are TVB-ready input data that can be obtained and used for brain research. So, for example, we have here the TVB time series and connectomes from tumor patients. So here's pre and post-operative data from 31 tumor patients and 11 controls. There's whole structure connectivity, functional connectivity, fMRI time series, and also the filled parameters of brain network modeling study with the tumor connectomes. Then we have the ADNI data, which is 33 participants that suffer from Alzheimer's or mild cognitive impairments or healthy controls. Here we have also the processed MRI, parcellated PET data, simulation results, services, EG, projection matrices to reproduce Stefanovsky et al. And from the Human Connectome project, we have data from 785 healthy participants, young participants, with their SC, FC, and fMRI time series in the Glaser parcellation, 379 regions, and pre-processed with the Human Connectome project's pipeline, which collects a number of best practices. Also briefly mentioning the Brain Atlas visualizer, which allows you to explore the 3D data sets. Okay, so now we are switching gears, kind of. So this is now the bridge towards the other platform, VRE, and mentioned previously, a big topic is basically making data reusable. And this is really a big problem because many studies simply cannot be reproduced because the data cannot be correctly interpreted or used. And the solution for this is to extensively annotate data with metadata, which is data about data. So this describes what is in the data. And ideally, this is in a machine readable format, such that these processes of discovery, management, use, and reuse can be automatized as far as possible. And metadata often makes use of an ontology, which is a controlled vocabulary to represent knowledge in a given context. So precise definitions of concepts so that we can make sure basically that we are talking about the same things. So and this is really the devil is in the detail often. So if someone says rain, but is then Graupel still rain or hail, and usually it's exactly these edge cases basically that are one of the central topics of the research. So it's really needed to have ontologies and to have a controlled way of talking about data and metadata. And this is all encapsulated in the eBrains knowledge graph, where we have exhaustive annotations. So all that we can see is basically encapsulated in a machine-readable format. And this format is JSON LD, or JSON or JSON LD. So JSON is the basic format, very similar to XML, for example. It is a data structure that allows to create key-value pairs, basically. So we have certain keys and map certain values to them. And by having these structural elements, we can basically express all kinds of data structures. So here, the problem is we don't know what refers this to. So what we don't want is to have redundancy and re-specify everything, but instead we link these different pieces of information. And this is really at the heart of what a knowledge graph is about. So there are different entities and they are all linked. And this is realized with JSON LD, because JSON LD extends this JSON with context. So here we always reference a certain resource that has a certain description of context. So context could basically be the rest of our knowledge graph, so of our structured knowledge. So knowledge is here really organized as a graph structure. So we have nodes and we have connection between nodes. And here such connections are realized basically. So JSON-LD is a format to store linked data, and it gives meaning to JSON using a context to interpret the properties and values. And this context also links the objects in a JSON document to concepts in an ontology. And with this, basically, there's a robust and interoperable and reusable, machine-readable way of expressing the contents of data, basically. So this can get arbitrarily deep. So we can basically nest these structures. So here's then a substructure, and all kinds of data structures can be modeled with such a way, basically. So you can have all you need is basically these curly braces, which contain then key value pairs, or here these other braces where you can have collections or lists of things, basically. And this is enough, basically, to specify data in a structured format, and also to automatically validate, basically, that the contents of a metadata are syntactically correct. So we cannot validate whether the contents, the semantics, are plausible. So I'm a string is it's just a string. So we know it's a string, this is good, but whether the contents of the string are plausible, we don't know. But at least we can validate the structural correctness. And in the context of Human Brain Project and of eBrains, there is the Open Minds initiative, which basically creates and provides metadata models for neuroscience research products like data, simulation results, computational models, software, data models. And the eBrains knowledge graph uses this language, so to say, to structure itself. So the knowledge graph is organized according to the structure given by such a schema, basically. And we have different types of schemas in Open Minds, like Open Minds Core to describe the origin, location, and content of research products, or SANS, which are schemas to describe atlases or anatomical anchoring. And we also have the controlled terms, which are all of these are regularly updated, and really this is a work in progress. It's currently at version 3, but it really it evolves and gets larger and larger, and integrated also with other neuroscience ontologies. And this is then an example how such a metadata this is an overview over all the schemas, basically. So these are all the schemas that we have in Open Minds. So we have actors, for example, where we can, so these are humans usually that we can specify with certain coordinates to describe them. And we have data. Here also we have different typical fields to describe them, or also all kinds of miscellaneous information. Then products, of course, very instance. So a typical product is always a data set, or actually a versioned data set, or a project, or a software, or a research product. And all of these can then be extensively specified. Here the research can be specified. For example, the specimen depicted in the research can be specified, parameters that were used, and so on. So this is a generic and abstract way to store metadata of neuroscience research. So for example, for persons, we can store, for example, digital identifiers, or contact information, family name, given name, affiliations, and so on, that we use to specify a person. The Open Minds team also provides the Open Minds generator, which is a Python software that makes it easier to generate these metadata. So we have in EBRAINS also the KG editor, the knowledge graph editor, which provides a nice GUI where you can also basically set the fields of a metadata set. But here you can also automatize this process. So for example, if you have 1,000 subjects, then you don't want to do this by hand, but write a script basically that generates the schema in a semi-automatic way. So you typically have some data sources that you then use to gather information, metadata, and then you fill the respective open-minds fields with this. And as trivial as they sound, but behind the reproducibility crisis that is often mentioned in scientific literature and also in media more and more, are behind these are the actual problems behind the reproducibility crisis. So data available upon reasonable request is something that we read often in papers, and the reality is that this data is never handed out, and basically there's always people always try to protect, or many people in the past tried to protect their data and stuff like that, and so this is a big problem for science. So basically, if the project is not reproducible, then it doesn't exist as such. So if no one else can use it and only you were able to use it, then it's not really a result. And this happens more often than not, actually. So when I run it on my machine, it gives me the following error. This is something that you read daily, basically. When you have a job in computational research, then this happens just always. Or that there are small differences between two digital objects that make all the difference, basically. Often it's really just one parameter value that makes all the difference. And it's not even like that the researcher wanted to hide something. It's just not easy to really think about every small detail and really integrate it and communicate it to the community. And therefore, there are more and more efforts to also automatize this process and to have it less rely on humans, which produce often errors, and have it more automatized. Okay, and this was already actually recognized quite early, I think, in 1995, and then there was a long break where no one really saw this, and then it became very apparent. So an article about computational science in a scientific publication is not the scholarship itself, it is merely advertising. So a paper is really just advertising. The actual scholarship is the complete software development environment and the complete set of instructions which generated the figures. So basically, if it's according to this definition, a research product is only then a research product if you can really trace the full chain from the very first raw data set that entered this research and until the final figures that are printed in a journal then. So this is basically the definition what really constitutes a computational science research product. And this was ignored for a long time, and now it cannot be ignored anymore and is now heavily addressed. And this is what Buckheit and Donaghy, I don't know what they actually were researching in '95. It was some geoscience or something. Actually, but in any case, they had a lot of foresight. So they said when they publish articles containing figures which were generated by computer, they also published a complete software environment which generates the figure. And this is really avant-garde at this time. So between this and now, there were many years where basically people did the opposite, basically. And one can define different levels of reproducibility. So what does it actually mean to reproduce a scientific result? So one aspect is that you can just rerun the same code in exactly the same way as reported. And this is often not doable. So often the code alone that is somewhere shared on GitHub or so is not sufficient. You also need some libraries and environment around this. So even if the code is there, it cannot be reused. Then now assume we have the environment and the code. We would like to rerun the same code, but this time we want to change aspects of it to assess, for example, its robustness, and to understand also what the code actually does. And this now requires much more. It requires clarity regarding the structure of inputs, outputs, and parameters. And this is really not trivial to communicate. I mean, here we can package it in Docker containers, which helped a lot, containerization. But here already we are at a stage, which has a lot of degrees of freedom, basically. And it gets even worse when we're going to this third level of reproducibility. So if we want to rerun the same model with a different implementation, then we really need an exhaustive specification of the equations, the functions, the algorithms that precisely and clearly communicate the theory from which any particular implementation follows. So the thing is that assuming there was some cool research with some code, then sometimes it also happens that you cannot reproduce this behavior when you implement your theory, your scientific theory, with different code. And then it turns out that the whole reason for your theory was not something on a high level, something theoretical, but it was just like an implementational choice, the way, for example, a random number generator was used and so on. There are many reasons that make you end up with a good looking result, but then if you try to reproduce it with an independent implementation, it suddenly doesn't work. And this is very important to really advance the research, basically. So often things just don't work if you re-implement them. And for this, you need a full and deep understanding of the whole theory, the whole ideas that are behind this research. And this is, obviously this was tried to communicate with papers, but papers fail at doing so. Also, communicating code fails at doing so. So there must be something else that communicates even more information. And we can learn a lot from the way that software is produced nowadays. So when big corporations, or also smaller, medium corporations, when they develop software collaboratively, they established a set of best practices that basically can be reused in science also. For reproducing workflows. So in code development, software development, many things are used that can be reused in science. For example, version control. So you all probably know GitHub and Git, where basically teams can develop code together, and you explicitly see what was changed at which time by which person. So really on a line by line basis, you can explicitly understand what happened to the code, how was it changed. And if we can have this for data sets also, this would be great, right? If every minuscule change to data set is basically recorded and tracked and can be understood later. And this is also called data provenance. So it's the history of digital objects. And the ideas here that basically come from software development is that to organize data sets in a hierarchy that can evolve, basically, and to organize data with standards so that this hierarchy is understandable by machines and can be reused. And of course, we mentioned this, annotate when described as metadata, to have these 3 levels of reproducibility. and also to package everything in the form of pipelines, basically. So to have automation as far as possible, and to have all these points where a pipeline connects to different steps, to have these automatized. Making the data accessible via web interfaces, or just easily accessible, and also making the code in computational environment reproducible software containers and virtual environments. So this we can all learn for research data management from the way that software is developed in big corporations. And this we can basically apply this logic. We can now not only apply to code as it is done in software development, but to everything, to raw data, to process data, to results, to figures, to texts, everything. And we can all organize them in a fair way, basically. So as mentioned, containerization is a great way to make workflow, to set up workflows, to specify workflows, to make them more or less platform independent. So you can easily share a container, and whether the receiver has a Mac or a supercomputer or whatever, they can instantly run this code. And the whole environment is already there and doesn't need to be installed. So it can be instantly executed. So this helps a lot for reproducibility. But even then, there are still often gaps, basically. And there comes data-led, which is a great solution, basically, and which is something I would think should be integrated in the future, probably in even operating systems, or it should be very tightly integrated into computing systems, because it extends this whole Git logic for data now. So if we remember, GitHub basically really tracks line by line additions, changes, and so on, which allows to really explicitly recreate what happened to a code file. Now we cannot do this for large data sets, right? So our data sets are terabyte large. So these data sets are large. So you cannot just, if you remove 2 GB, you cannot just store this in a Git repository because it's just too much information. This would accumulate, and this would be huge, huge data sets. Therefore, here only a hash of the large file is computed. So this is like a hash can be seen like a mean value of all values in a data file. And this basically allows us really to version control and track the provenance of arbitrarily large files, and to extend this whole Git logic now to all kinds of files, to every object that is in a computer, basically. And this also makes it easier to share and obtain data and to reproduce workflows. And it's completely domain agnostic, so it doesn't matter what you do, what kind of research, for example. It also allows others to recreate or reproduce the research without actually knowing what happened there. So upon button press, basically. And data-led data sets are nothing else than directories that are managed by data-led, and these are nothing else than Git repositories. And Git repositories, basically they store the data at a safe place, replace them by empty files such that they cannot be overwritten with write protection, and really manage the way a data object is processed on a computer. So basically the workflow looks like you create an empty data set. So this just creates this initializes a folder and creates some initial metadata. And then every time you want to record the state of a data set to history, then you can just run such a save command. And with this, create a snapshot of the local state, basically. And also you can extensively specify where the sources of input are located, where it comes from, and really also store the full data that is necessary to recreate a result. And this can be done in this nice nested way. So as mentioned, Git really just just adds these hidden folders basically where metadata about the actual data is stored. And then we can basically store the full containerized workflow and the data, and then we can nest this. Basically by having subfolders, one can have a modular structure basically, and reuse components in different analysis streams, for example. And this is, it's a really just nested folders. And the cool thing is that DataLad always keeps track where the actual data is located and where and only pulls it when it is needed. So these data sets are not massively large, right? They don't accumulate and get larger and larger and larger as you work, but DataLad takes care that only the crucial data is stored only once and is pulled if necessary. So this is really cool. And with this, you can record where you get something, where it is now, and basically what you did and how you changed it. And it captures the whole provenance information, and also allows only to overwrite files if you specify this. So with this, you can ensure that the whole workflow happened in a reproducible way, and have all these metadata stored that allows you to reproduce the workflow. And basically, even without knowing what happened in the workflow, you can just look at the commit messages, look up the commit code, and then just reset a data set to a historic state, for example, or rerun, for example, steps, or reproduce and verify results. And this granularity can be freely decided. So it's really domain agnostic. It's every change that happens to a file on a computer can be tracked in this way. Okay, and so this was about data provenance and making data reproducible. And now we would look how this and other things are implemented in the virtual research environment, which is a platform of the Charité, where researchers can process data in a secure manner. I think I have a video here. I will probably just go through the main parts of this video with you. So this is the landing page of the virtual research environment. This happens when you just go into the URL of the VRE, and there you can see then a bit of information about the VRE, its partners who build it, a bit of information how much it is currently used. And then you can basically log in. Here's you have a password and a login username, and then you can log in. And then you are greeted by this projects overview page, and you can then select your project that you want to work on. So a project is basically your isolated space where you, together with your team, can work on a data set. So here we see some statistics. Here we see a recent file stream. Here's a file explorer basically that allows to have an overview over what is stored in a given project basically. We can also search this file stream, specifying, for example, dates and so on. And here we have our file explorer. So this is very similar to the eBRAINS Drive, basically, where users or teams get a file system in the cloud, which they can operate on. They can upload files there. And the important thing here is that the upload happens in a coordinated way. So you cannot simply upload data, but you upload the data first to the so-called green room, which is a safe staging area where the data is securely brought into a form where it can then be processed in a GDPR compliant manner. So only then this copy to core is possible, but only possible for certain team members. And here then data sets can also be created. So there's this concept of the VRE data set where collections of data can be created. And then we can annotate this data. We can give it a certain structure also, and also annotated metadata. So here we see that we have given the data a certain structure, and we can then validate the structure. It's the bits format, and we can see whether the structure of the data adheres to a certain format. And here we can then annotate the data with metadata. The VRE has an own schema, but you can also use the OpenMind schema or your own schemas that you want to create. So you can basically create all schemas that you want, but of course it makes sense to use existing schemas that also others understand, basically. And with this, you can then really recapitulate the history and what happened to a data set, basically. And you can also then make releases of a data set. It also happens automatically when you say now is a good time for creating a snapshot, basically. And can then release the data and have it in a fixed, immutable form, basically. Here we have a extensive search functionality, so we can combine all kinds of query elements to extensively search such a project. So all that we can see here in this left sidebar basically belongs to this specific project. And a user can, of course, be a member of many different projects and can, depending on the project, do different things, have different roles, for example, and so on. Then you can also communicate with your team members with different ways. So announcement is quite a strong way that you really want to make sure that the signal arrives. And but there's also the XWiki down here, which will come later, where we can extensively document things. This is configuration of the project. Some general configuration options. Here we can request that a certain data set is copied from the green room to the core. So this request is then made against a person who has basically the permission to copy this data, and this is also has the legal role, basically. Apache Superset is a business intelligence tool which is also integrated here. Guacamole is a remote desktop interface, which is really cool because there you can basically, you have really as a team your own computing space in the cloud. So you have a computer in the cloud which everyone can use, a virtual computer. You can have as many virtual computers as you want. Not really. You can have a certain amount of virtual computers, and basically then process data with your team in a secure manner. Of course, also Jupyter Hub is there, which is widely used, and XWiki is then a nice solution for documenting the research results, basically. So here, basically, you have the full wiki functionality that you can then adapt according to your needs, basically. Okay. This was most of the video. There's also extensive documentation, and you can also submit support requests, and there's also, as mentioned, extensive user guides and documentation. Okay, this was an overview over the VRE, and now to summarize this, what is the purpose again of the VRE? So it's a data management cloud platform for biomedical research that protects the privacy of research participants. And it allows to store, process, and share data in compliance with the European Union GDPR. Importantly, it reduces the barriers to biomedical research by addressing a lack of infrastructure that can protect sensitive data, can process complex data, like MRI, that allows to create custom processing workflows. So this is also a big topic here. So there are platforms online, but either they provide no infrastructure at all, which makes it hard for users to get a foot on the ground, or the infrastructure and the software and the services are so specialized that no one really, or only a few people can really use them for their research. So really having this balance between a platform that gives you certain tools at hand that you can use for research, while at the same time providing all the flexibility and basically allowing you to do everything that you want, it's quite a balance act, as you can imagine, due to data protection, obviously. Lastly, one of the main purposes is also to provide access to high performance computing. And here the same in a more visual way and from a slightly different perspective. So the VRE really tries to balance these highly different needs, having security at the one hand and full flexibility at the other hand. And it does it by offering graphical user interfaces, which is welcomed by a lot of users, but also command line interfaces where you can really automatize and program your workflows exactly as you are used to it from your local computers and local machines. And importantly, you have the opportunity to process data in the cloud in a private manner. So you have your team, you and your team are together, and you really get direct access to chips and to storage, to CPU and to storage, without anyone else being able to access this, where you really can privately work on your research, but with extensive resources and together with others. And all of this in a GDPR ready way. So it was certified also, and is also integrated in the critical infrastructure of the Charité IT network, which adheres to, it's a certified critical infrastructure of the German government, and adheres to a number of SOP, standard operating procedures, to ensure this protection, basically. Another big aspect is, since this, we're at the Charité, obviously a big hospital, so we want to automatically ingest also the data from the bedside into the cloud. So the Charité, for example, has PACS servers where all the PACS is a common format for storing patient data. And these are also connected to VRE, and new PACS data can be easily automatically scraped and ingested into VRE projects. And all of this, together with metadata and standardization, provenance tracking, version control, is to ensure a fair usage of data and scientific process. And this logic basically is now also extended towards EBRAINS with the Health Data Cloud, where similar services like provided by the VRE are then implemented and deployed in the EBRAINS infrastructure, to have really a federated research data ecosystem that enables neuroscience research consortia across Europe and beyond to collect, process, and share sensitive data in a GDPR compliant manner, with seamless access to supercomputers, interoperable with metadata, and built on the best practices of cloud computing, basically, that also provides agility and scalability. These fair data needs, I think we can skip over this. So again, extensive metadata capturing, annotation, provenance tracking, support for open standards. You can quickly go over this. So we mentioned that the basic goal is that data is reusable and that systems are interoperable, basically, which is the problem everywhere with digitalization in many different areas. So a more schematic overview over the VRE. We already heard it. There's this green room which provides only restricted access. So when data lands first, when data comes into the VRE, it first lands in the green room, whether it comes from hospital data sources or uploaded by a person. There really the data can be pseudonymized and prepared and minimized, such that the whole team can work on this data in a compliant manner. So for example, here, sensitive information like, so here basically everything that is done to minimize the data set to the minimal state that is needed for research, such things that we mentioned earlier, like this bringing together of genomes and addresses, is prevented. So the architecture is, we have the research portal with a GUI and a command line interface to manage these resources. And we have the separated zones, green room for data onboarding, and the core zone for processing the data. We have storages in the background that allow to store structured and unstructured data and to search this data. It can then be annotated as metadata and version controlled. In the backend, there are high-performance computers and, importantly, also virtual machines with remote desktops and command lines. So your team gets their own virtual machine where they can process the data together. And this is all modular and extensible. It's a service-oriented container ecosystem connected with RESTful APIs and orchestrated by Kubernetes, and with automation as far as possible. Yeah, we saw this in a video already. We can create these collections. So these are virtual folders to group files without affecting the storage location or data lineage. So this is just a personal way of organizing data. But the more important way, as we mentioned, is this data set concept. So a data set is basically, the idea behind this is that data is organized according to a data standard. So BITS, for example, brain imaging data structure, is a schema that describes how the actual data files like MRI data, EEG data, and so on, how they should be organized and how the file names should be called and so on. And then the second important ingredient is this metadata annotation with JSON, JSON-LD, using the open minds schemas and mapped to neuroscience ontologies. And also the ability to make these immutable snapshots or versions, basically, where we can be sure that we are talking about the same thing. So there's an identifier that really uniquely identifies basically a data set where we can be sure that yeah, we are really accessing and talking about the right thing, basically. So we saw this already. We can create data sets, we can annotate them, we can verify their correctness, we can then release new versions. We can also inspect them, download them, edit them, delete them, validate the structural correctness. So here was an error. This is a video. All right, so this is this shows the whole dataset concept again. So dataset is an orthogonal concept to projects. So datasets here, you really here have basically these immutable versions, or these structured versions that are used to communicate results with others. Metadata annotation, again, we use JSON here. We saw that already, how JSON is structured, again, with open minds in the background. And here we have then the opportunity in the VRE to either directly use the open mind schema, upload our schemas, or to also create our custom schemas if we want to invent a new metadata standard, basically. And this allows them to track all this provenance information. So here we can exactly see what happened basically to a file, to every file, and at a certain point create releases. And this creates us then a lineage of data, of digital objects, basically. So we know exactly then always the current file, which processing operation happened to it, and from which source file does it originate, or how did it come into the VRE. So we have extensive properties and attributes annotated and added to each file that allows us then to understand the lineage of an object, and basically for every file. So this is now a way to visualize this, but under the hood, basically all of this is structured in a machine readable format that makes it easy for other platforms to use this and work on this. As we mentioned, the green room is the only opening or door through which the data can enter the VRE, and it's a staging area to minimize the data. So here we can really follow the principle of purpose limitation and data minimization from Gdpr, because then only the project administrator can authorize the copy to core and make the data available to the entire team for specific purposes. And only certain people really have the lawful basis to make these operations and to instruct others to process the data, which is all mapped onto this technological workflow, basically. So this organizational request from the GDPR is here mapped on technological solutions, basically. And data can be uploaded via GUI, but also via command line interfaces or automatic pipelines. So you can also, nevertheless, create scripts that automatize this process. The core zone is then really the main data processing arena, and we have now seen it extensively, where you can make all your processing that you want. The utility zone is another aspect of the VRE, and these are basically back-end services that support all these front-end services and that work under the hood or behind the scenes, so to say, in order to ensure this data protection, for example. So these utilities then ensure a managed way of copying data such that no others can access them, and so on. And there are also continuous integration, continuous deployment runners that constantly basically make sure that the whole system is always up to date and always works and is stable. Then the Workbench. So this is then really the heart where researchers will mostly be. So here you have basically the same that you have when you have a computer at home, but this time in the internet and with a few more cores and with a bit more memory, such that you can really also compute more sophisticated or resource demanding workflows with your team. But again, there's also, of course, the high performance computing backend. So if 24 cores and 128 GB of memory is not enough, then you can have hundreds and thousands of cores basically on the HPC. Jupyter Hub, a favorite of many. Many like to use Jupyter for interactive processing. It's also really nice to create code and make exploratory research. So this is obviously a feature of the VRE. And the command line interface now, if you want to really automate things, gives you powerful functionalities. So basically the whole functionality of the Linux terminal and other VRE specific functionalities. So for example, you can create cron jobs that automatically do certain things at certain times, or automatically scan for newly incoming data, or automatically upload data, or automatically pre-process data. The command line interface lets you basically interact with the VRE, so it lets you do everything with the VRE that you can also do with the GUI and even more. So here you have the whole basically with simple commands, you can operate the whole VRE and operate files, just like if it would be your local computer. You have here access to a whole platform basically that can be configured via command line commands, and even the HPC backend can be configured and submitted and queried with this command line interface. So this is a really useful tool. For example, if you want to synchronize your local file system, this is quite practical. You don't have to like always make sure that you copy individual files back and forth, but it just automatically creates the same state locally and in the cloud. Few words on high performance computing. Currently we have 200 nodes in the Charité, IT for research, HPC for research environment. So each has 16 cores and up to 192 gigabytes of memory. It's also some high memory nodes, a lot of storage for fast processing, and also some GPUs. And this is intended really for large-scale automated workflows, very much in contrast to Jupyter Hub and also to the virtual machines, which are more for normal compute operations. Again, processing of health data is generally prohibited in the EU, but the VRE offers privacy by design and by default, which really makes it possible for controllers to comply with the law, because the whole organizational setup of the VRE allows that the controllers always stay in control, basically, and do not lose this control. And this is implemented by having different user roles, basically, with different permissions. I think I will now skip over most of the rest. I think we are almost at the end, so I will just briefly go over a few things and then have some time for questions and then end the session. We mentioned this, the VRE is embedded in the critical infrastructure of the German government Charité IT, and therefore implements best practices for security and data protection. So for example, what are these practices? The direct access, as we saw, is not possible. But only a controlled and locked access that can also be then basically a record exists. Basically, a record of user activities exists, which can also be then used to demonstrate compliance with GDPR. And we have different namespaces and zones and multi-step protocols, basically to isolate these different concerns. And we mentioned this. So with this kind of data, really, even after directly identifying information like names, faces, addresses, and so on were removed, which is often called pseudonymization, the data is still considered as personal data under GDPR. And therefore, as we've seen earlier, it's really easy to leak sensitive information out, and it gets easier and easier. So here the responsibility is clearly defined, and it's clear who has which responsibility to demonstrate this lawfulness and compliance. Of course, data is encrypted during transmission to and from the VRE and also at rest while it's stored on a disk. Technical authentication and authorization is also another technical measure, basically to ensure that only authorized people can access this data. So currently, to access the VRE, you have to provide a personal identification and evidence of a contractual relationship in order to create an account, basically. fine-grained and role-based and project-based access control is another feature to demonstrate compliance. And with this, I think we are now at the end.